Welcome back to the final talk in this morning's session, and it's a pleasure to welcome Alf Gerisch from Technische Universität Darmstadt, and this morning Alf's going to be talking about non-local models for interaction-driven cell movement. Alf.
Thank you very much, Mark, for the introduction, and thank you very much to the organizers of the workshop for the opportunity to present here, and of course also thank you very much to the organizers of the whole program, in particular Untitled, for putting so many great events together. Okay, I'm going to speak about non-local models for interaction-driven cell movement, and this is joint work I've been doing with Kevin Painter, Jim Bloomfield, and Jonathan Sharrett from Harriet Watt in Edinburgh. And what I'm going to speak about is first some biological background on cell contact. So what do we put in our model, which I will then formulate in a second part, and we will have a look at what kind of patterns are possible with the models we will look at, and we cannot do this in full detail, so we have some exploratory analysis, and this model formulation and pattern formation investigations, they will be done for a model where we have just one cell type, and then also for a model where we have two different cell types possibly interacting. And application-wise, I will look at what is the potential of these mechanisms, which we will formulate in the model on, for instance, the dispersal of neural crest cells, and in the second example, the organization of pigmentation stripes in zebrafish. Okay, cells communicate with each other in various ways, and one way they can communicate is via direct contact, so in particular membrane molecular binding, adhesion we have seen, and exchange of information through such direct contact. And we will focus on direct contact here exclusively, as it is, for instance, given in cell-cell adhesion. What we do not consider here, because we want to focus on the first point, is indirect contacts. You can also have communication between cells via indirect means. For instance, in chemotaxis, the signaling is between cells is mediated by a diffusible chemical. So we do not look at this here. Okay, so this communication via direct contacts that can occur on very short distance, but it can also occur on a rather long distance, so up to 50 cell diameters has been reported, and this long distance communication, if you want, this then happens via cell protrusions, for instance filopodia or cytonemes, which can really extend far away from the main cell body. And as we've seen in the past week, and we all know, the reality is full of nice detail, of interesting detail, and obviously also of nasty detail. And in particular, cells are complex, they are morphing objects with lots of structure and chemistry. And in our model, we will abstract from that quite a bit and consider a rather generic setting and see what we can still understand of such a model. And one of the consequences of direct contact is, for instance, movement. This is what we will focus on here, but contact, of course, can also lead to many other responses. Okay, so instructing others to move is fundamental for many animal or cellular populations. We've seen in the first talk this morning examples of flocking, and so there we look at animal populations. We will concentrate rather on cellular populations, and but maybe this is not such a huge difference. Okay, so commands to instruct others to move, they are transmitted by contacts over variable distances, so they can be short or long range. So here, for instance, we have two different cell types, light one and a darker one, and you see, for instance, that the light ones, they communicate over a very long distance, whereas, for instance, the darker ones, they might just be able to communicate over a shorter range with each other, because they cannot get in contact over longer range. The effect, these commands, they can affect cells of the same type, so just the dark cells or just the white cells. So in that case, we speak of homotypic interactions, or they can affect cells of different cell types. In that case, we speak of heterotypic interactions. And so, in particular, it is not necessary that the impact of the dark gray cells in communication with the light cells is the same as the impact on the light cells when communicating with the dark. So there might be some asymmetry, and we'll see an example of that briefly. So these mechanisms of movement, they can have a significant impact on the organization of tissue, of course. And the first example I want to give is cell adhesion. And this is determined by the expression and regulation of cell adhesion molecules, so transmembrane molecules with a intracellular part and an extracellular part, here connected to the actin cytoskeleton. We have seen many examples of this during the week, and the extracellular part, extracellular domain, can bind to corresponding catarrhine molecules on the neighboring cell, and so we have a connection. So catarrhines are mainly involved in cell-cell adhesions. They're integrins involved in cell matrix adhesion. There are a few others. But in a sense, the cell-cell and also cell matrix adhesion is crucial for tissue and organ integrity. And yeah, we see an example of some cell-cell contacts forming, and the three cells, they get nicely together and are tightly bound in the end. And if I can adhere somewhere, then I can also exert a force there and pull myself along. So more or less this static property of tissue integrity or organ integrity, which goes along with cell adhesion, also has a dynamic component, as it allows cells to move. Okay, and this is what we see here. Yeah?
May I interrupt you? You made a statement now. You said that if I cannot hear, I can exert also force. You include, and pull myself, you include also the case of friction, I suppose. So resist to movement, not only the forces that I actively exert. That is, you assume that there is some myosin there, or something like that.
Yeah. Well, this is one option. If you can hold on to somewhere, you can exert force, and this is translated into movement. And there might be friction involved. Okay. So here we see an experiment by Putti and Steinberg, where they have cells in a petri dish, and the N-catarrhine expression on the cell surface is increased from A to B to C to D. And what happens if you let these cells just sit there for some time? They start to aggregate, and they aggregate in particular into larger and larger clusters. And if you have two different cell types, which are sort of dissolved here and then mixed in a pellet, then if you wait some time, then if they have the proper adhesive properties, then these two cell types, they sort out again. The one with the stronger adhesive properties sorts to the middle, the one with the weaker ones to the boundary, and we get the cell sorting behavior. And there's the differential adhesion hypothesis put forward by Steinberg, and which says that the final state of such an experiment just depends on the self and cross adhesion coefficients, which are associated with the two cell types here. So this was adhesion. So cells more or less like to aggregate with each other, But there are also certain cell types which repel each other on contact, so contact inhibition of movement, and this has, for instance, been observed in neural crest directional migration. So here we have two cells which are moving to each other. At some point they are in contact and they break down the contact immediately again and move away in different direction. That's crucial for these neural crest cells because they have to disperse in a tissue and they should not aggregate, and we'll see that later on again. Finally, if we look at mixed populations, we can look at cells which are in the implied in the pigmentation of the zebrafish. So we have melanophores and xanthophores, so these are yellowish, those ones more blackish, and if we look at these, then here we see these cells, they like each other quite well, so they don't necessarily run away, and whereas here the xanthophores, there's a little bit more of dispersal, but yeah, we see here it's a nice spacing. But what is even more interesting is if you put melanophores and xanthophores into one thing, then the yellowish type is sort of chasing the blackish type, and the blackish type wants to go away, but no. Yeah, and you can also see this nicely here. So
It's bigger. Size doesn't matter.
okay, so we have this attracting effect of cell contact, we have repelling effect of cell contact, and we have interaction, positive or negative, attracting or repelling between different cell types.
Do you know what these cells are? The different types of cells?
It's pigmentation cells in zebrafish, and I'll come back to that in the end. Okay, if you want to model this, you could use, we've seen individual cells, so you can use discrete or individual based models. Each cell is defined separately. We can treat small scale problems with this, with relatively few cells. And there are many approaches: cellular POTS models, cellular automata approaches, deformable ellipsoids, lattice-free approaches, immerse boundary approaches, many, many more. I've given some references without any, I don't think that's anywhere complete because it's so much being done, and we have seen a lot of this during the week here as well. So these discrete models, they're typically quite simulation heavy. Analysis is often difficult or limited. Somehow they're also implementation dependent, and the big question is sometimes how to compare, maybe to experimental data, but also to different methods between themselves. So I have not seen much of comparing these different approaches. So we will focus, so here's an example, thanks to Kevin Painter, of a very simple agent-based modeling for cell adhesion, differential cell adhesion, and you start with something and then you end up with a mixture of cells, yellow and red cells with different properties, and depending on the properties you get different sorting behavior, the right one being just a homogeneous mixture. In continuous models, on the other hand, we do not look at individual cells anymore, but the cells are described as a continuous cell density distribution, so we don't see individual cells anymore, but at any place there's a density. Okay, and that's why then typically PDE models, we look at larger scale models, cancer, tissues, maybe even whole organs, and how can we, for instance, put sort of adhesive effects into such models? And one option would be, or which has been used, would be a to add or to go from a standard Fickian diffusion to a cell density dependent diffusion coefficient, and you would choose a decreasing function d of u for your diffusion coefficient in order to represent greater adhesion. And with that, you can, if you have an sort of a concentrate or a lump of cells somewhere in your spatial domain, then with such a model, you can ensure that it does not diffuse out to every other space position in your domain, which would be the case with standard diffusion, so it's limited. But what models like this cannot explain is a spontaneous aggregation. So you have a uniform distribution of your cells, and suddenly at some point an aggregation is triggered, and also cell sorting cannot be modeled with such a model. We've seen many examples of chemotaxis, haptotaxis this week. So one could think of, okay, if I want to aggregate with my peers, then I move up gradient of my own density. So one could write down something like a self-taxis type process by adding such a taxis term here, where the gradient is of the function itself. And with such a model, yeah, pattern formation is possible, but if the zeta becomes too large, and that would be the interesting case, then we have a backward diffusion problem, and yeah, that's not well posed. So that's maybe not really the way to go with thought. And if you look at discrete models, then they always retain some degree of non-locality in order to model attraction. And this is what we also want to do, and we have seen this in the talk this morning. There was also a non-local term. Okay, so we look at now at our continuous model, one cell population, and the cell density is denoted by u, depending on space x and time t. We follow a mass conservation approach, so we don't want to lose cell mass or just create it anywhere. And our focus will be on cell movement driven by direct cell contacts. Everything else is deliberately kept simple, and in a real model, these two things could be much more complicated. So we look at Fickian diffusion simply, the term you can see here, and a logistic growth, as you can see here. But the interesting term which we want to study is this one here, and I will explain this a bit more on the following slide. But this is the model which we study after non-dimensionalization, and then the proliferation rate rho is positive, and the carrying capacity after the scaling we've chosen is between 0 and 1. This is a little bit important later. So we want to focus on this term here, and let me first explain the parameters. We have with mu the interaction strength, and if mu is positive, we will have an attractive interaction between the cells, and if it is negative, we have a repulsive interaction between the cells. And we have a parameter xi, which corresponds to an interaction range. So if xi is larger, this means the cells can sense their environment in an even larger space, and as I said earlier, up to 50 cell diameters, that's even possible. We normally don't go that far, but it could be done. Then we have a function g of u under the integral, and we consider here the very simple case g of u equals u, and this in essence means that with increasing cell density, I have a higher chance of making contacts to other cells, and thus I have a stronger interaction. That's a very simple assumption, but this is the one we've used here. For practical models, the saturating or bounded functions may be more plausible. we have this, after non-dimensionalization, this prefactor 1 minus u in front of the integral, and this is a factor which limits movement by adhesion, and so it's something like a volume filling prefactor, so it should ensure that our cell density stays bounded, even so it's not aggregating arbitrarily high. And then we also have here a so-called normalized interaction function, omega tilde, which says how much does the distance from I'm the cell, I'm sensing that region, how much does this distance influence the interaction? And so that's a convolution. That's the convolution kernel, omega tilde, and we've used a couple of, considered 3 of them. I will say something in a minute. Yesterday there was the question why this is a velocity, and so this in fact is a vector. We have the distance s from the cell where I am to where I'm sensing, normalized with the absolute value of s. So this it really gives a direction, and it's a force. Yeah, you can derive it from Stokes' law, and
it's a gradient of a potential.
or like this. Yeah.
That's not the form.
Yeah, you can assume you can look at Stokes' law of a spherical object pulled through a viscous fluid, and then you arrive an expression like this. And then you sum up overall your sensing region. Okay, so we looked at 3 different normalized interaction functions. One is just, so it's r over c is the argument. So if r equals c, the interaction range, this is sort of a critical, then r over c equals 1, and this is where changes normally happen. So this moving?
Depending on that.
Okay. It's attracting. Yeah, we have this simple step function equal to 1 if the ratio is below 1, and equal to 0 if the ratio is larger. We have an exponential decaying profile, and then we also have this hump-shaped function where the maximum is at r over xi, and so with this function we can tune how far away from the cell the best attraction is used, and I think this kernel is also used in flocking models quite happily. Why we've also chosen this form, because then later in linear stability analysis we then see that the non-local term can be read as a Fourier transform, and this Fourier transform for these functions is simply known, and so we can really write it down and go on with the analysis. Okay, if we look at our one cell population model again, then there are uniform steady states. If I have a uniform solution, then this integral always averages to 0 or evaluates to 0. So the steady states are dictated by the logistic growth in our case, and if there is growth rho greater than 0, then we have a positive uniform steady state U, and if rho equals 0, no cell proliferation, then the steady state is just given by the average initial cell density, which we will also refer to as capital U. And so our steady state is capital U, and then we can perform a standard stability analysis. So we perturb the steady state with some small function U tilde, and then we look for solutions U tilde of this form, spatial mode times continuous in the time with amplification factor lambda. We insert this into the equation up here, this u linearize, and then we obtain lambda as a function of k and the parameters of the model, and the necessary condition for the steady state to be unstable to spatially inhomogeneous perturbation is that for some k, the real part of the corresponding lambda is positive. And if you do the calculation, then this necessary condition can be transformed into this condition, where capital gamma comes from the Fourier transformation. And it holds that it is positive for all k greater than 0. And what we see from this condition is, so u is between 0 and 1, so this is all positive. Mu can be positive or negative. This was our interaction strength, repelling or attracting behavior. Gamma is positive, so if mu, and on the right hand side, we have just a positive number, so but if I choose mu sufficiently large, sufficiently attractive behavior, then we should be able to see thus instability arising and in fact aggregation. And the instability is not possible if this interaction parameter is negative, if I have repelling interactions. So then it's always going back to the uniform steady state. And for this second function omega tilde, we can in fact perform the analysis explicitly, and we come to this precise condition here. And graphically, that's easier seen. And so this is the analytic result, and for the first and the third function, we've just evaluated numerically. And what we see in the top row is the stability instability diagram without cell proliferation, and we see if mu gets large enough, then we can expect patterns. So this is the colored region. If mu is too small or xi is too small, then we're in this white region, and this is where the necessary condition is not satisfied, so we certainly have stability of the homogeneous steady state. It looks a bit different if we switch on cell proliferation with rho equal to 1. And here we can also have the case that we start without pattern and increase the interaction range, and if we increase further, we leave the pattern domain again. And the color code is the expected spatial wavelength, so the wavelength corresponding to the maximum lambda value. And if the interaction range xi increases, then the wavelength becomes bigger, and this is probably also expected. And we can do simulations to confirm what we've seen in the diagrams. And so what we see here is so space is the x-coordinate is in the horizontal direction is always space. In this top row plot, the y-coordinate, that's the time from 0 to normalized 200, and so that's a space-time plot with black means large and white means low values. And at time t equals 100, we plot here the cell density over space. So at this line, this is the actual profile which you obtain. And what we observe is this is for a fixed interaction range xi equal to 1, and we increase from left to right the parameter mu, the interaction intensity. And as soon as this one is large enough, we go from uniform steady state to patterned solution. And if we increase further, then the pattern becomes of smaller wavelength. So this is somewhere if you go up here in this direction, starting down here, and you move up, and you see then it goes from red to blue, to wavelength becomes smaller. Here we do the opposite way. We go from left to right, and there the wavelength becomes larger. We have a fixed interaction strength, mu equals 5 here, and then increase the interaction range, and you really see bigger aggregates. If you switch on cell proliferation, all gets a little bit more dynamic. We have emerging and of peaks, we have merging of peaks, and so on. And but the principle is more or less the same. Here we see the case. We have a fixed interaction strength, mu equal to 10, and increase the interaction radius. So we go from from homogeneous solution to patterned solution. And if we increase too much, we go back to a homogeneous solution. So that's all in line with the analysis. And this can then be taken to 2 dimensions with 2 cell distributions, U and V. We have enough parameters anyway, so we do not consider cell proliferation here anymore. And after non-dimensionalization, we obtain this system of integral differential equations. The diffusion coefficient of the 2 cell population, that might be different. Here it's normalized to 1, and here we have a d which might be not equal to 1. The volume filling term now becomes 1 minus u minus v, because u and v take up space. And we have for each density 2 of these integrals, 2 interaction terms, interaction with itself and interaction with the other cell cell type. Yeah, here and here, but the form of these, it's always the same, and but we can have different strengths for each of the interactions and different interaction radii if we want. And since we have no proliferation, the uniform steady state is the initial average cell density, and capital U, capital V, and we assume that this is the sum is somewhere in between 0 and 1 after normalization. Good, and then we can do linear stability analysis again. We look for solutions of this kind with these kind of perturbations, and this leads to a quadratic equation you can solve. I think there's a bracket missing, and for instability now it is necessary that the real part of some eigenvalue is larger than, or of the larger eigenvalue is larger than 0 for some k, and this is equivalent to this coefficient c of k or the coefficient d of k, which are in this quadratic equation here, being negative for some k. And the expression for c and d are, well, not the nicest ones, but if you look at them carefully, then you can obtain the following table. So here we have the homotypic interactions of u and v. So here it's both are self-attracting. Here u is, one is attracting, the other one is repelling, and here is u and v are self-repelling, and these are the heterotypic attractions, u to v and v to u, and again this can take one of the 3 cases seen here, and if there's a tick mark, that means in principle there are parameters which should allow for pattern formation. And there's only one case down here where we can say under no conditions will there be any pattern. And this is the case if the 2 cell types repel each self, themselves, repel, and the first cell type is attracted by the second, and the second is repelled by the first. So that's the only case where no patterning is possible. And a more detailed analysis is quite intricate, because we have quite a number, 8 parameters, if you like. And you can have a look at certain situations which are biological meaningful, and then do an investigation and not of the full parameter space. And so we can plot or generate plots like this and investigate the numerically the pattern formation potential. Not going to discuss this in all detail here, but this set of simulation corresponds to the table which you've just seen, and bottom row in the middle, that's the only case where no patterning is possible. In all other cases we can have patterning. Here's the all repelling case, and we get a nicely separated pattern of U and V. And
this is all in one space dimension, is that right?
the simulation is in one space dimension. Yeah.
And the theorem?
Oh, the theorem is also in one. Yeah.
I wonder if more than one makes a difference, or just more complicated.
I don't know. We haven't looked. It was enough parameters so far. Okay. Well, it wouldn't be more parameters necessarily in 2D, but yeah. So we see patterns where sort of the two cell population mix and form clusters. We see patterns where they separate, form clusters. Here they have the same height. Here they have a considerably different height, and if you think about it, then it can all be tied down to the values, whether it's attracting or repelling in the appropriate places. So for instance here, one cell type is repelled by the others, the blue one, so that one tries to move away, but on the other hand the red ones like the blue, so they pull a few of them in, so you have a low density here, and yeah, both cell types are self-attracting. That's why you get these clusters, but you get these height differences. So there are different situations, and yeah, how can we apply this to some biological situation? And the first one is the dispersal of neural crest cells, and something which happens during embryogenesis, and the neural crest develops along dorsal midline, and it's crucial for the development of the nervous system, for skin, skeleton, and so on. And what happens is neural crest cells detach from the neural tube, and they migrate in precise route to their respective destinations. So some move here, you see, some move down here, a few split up there, and the others go further down. And that's a well orchestrated process where probably many factors contribute. So not only what we will consider in the following, but the failure of these neural crest cell populations to disperse and colonize to the intended target tissues, that gives rise to birth defects, and I've spared you some pictures here. And in view of the approaching lunch, and in general, neural crest cell dispersal is also an important case study for understanding cancer invasion and other pathologies, because we heard about EMT yesterday, and this is something which also takes place here in a physiological situation, and which can be studied apart from in a tumor, for instance. Okay, there are various hypotheses for how this dispersion works. So, for instance, random movement plus proliferation has been put forward, guidance in response to attractive repelling chemical cues, so chemotaxis-like mechanisms. And it has also been proposed that contact inhibition of movement may lead to this suitable dispersal of the cells, and this is what we want to concentrate on, see is this possible or not. And we have seen in a picture earlier on that these neural crest cells, they sometimes come in contact, collision, they have a contact, and then these protrusions collapse, and the cells change direction, then they move away in a different direction. So in this crest cell dispersal, that more or less happens on sort of sheets, and you have leading cells and trailing cells, and in effect one could consider this in a very simplified situation as a quasi one-dimensional problem. With one repulsive cell population, we assume logistic cell proliferation and reasonable parameters from the literature, and we have an initial compact population in a, if you like, a ridge, which corresponds to the neural tube, and then we put all this in our model and ask how do the interaction range and strength of repulsion impact on the dispersal process. And what we see is from at x equals 0, we have this initial ridge of cells, and we get nice traveling wave solutions with speed increasing, with increasing interaction strength from left to right here, and with speed increasing with increasing interaction range. And the different lines you here see are snapshots of the solution at different time points, and the cells are moving outward in a more or less traveling wave fashion. And you can estimate the invasion depth and then the wave speed. And what you observe is where this is dependent on the product of mu and psi only. So we can have them as one parameter, and it would be nice to have a analytic prediction of this wave speed. For instance, we haven't done that, but I guess a traveling wave analysis could shed some light on there. So what we're not saying is this contact inhibition induced repulsion is the mechanism which explains the dispersal of neural crest cells, but it could be one possible explanation. And this is all what this simple experiment demonstrates here. The second application we looked at was zebrafish pigmentation patterning. This was the run and chase of the cells. So there we have, in zebrafish, we have this stripe, interstripe pattern, and between the yellowish white and the black, there is again also a thin separating strip which you do not see without any pigment cells, but in the blackish stripes there are melanophores which are black, and that will be cell density U in the model, and we have also some light reflecting iridophores. That's why it's so nice and bright. And in the whitish yellowish stripes there, the pigment cells are xanthophores, and again some light reflecting iridophores. So they are everywhere, these iridophores, but they are not in our model, because initially it was thought that only these two are responsible. The viewpoint is changing slightly. So we have cell density U and V, and there have been Turing-type morphogene-based models in the 90s to explain this patterning, and the pattern looks nice, but so far no chemical morphogenes were found in these zebrafish. And what we follow here is a minimal set of pattern-generating interactions, taken more or less from this paper of 2014, and this run and chase scenario, one cell type attracts the other cell type is repelled from the first one. That was proposed to explain pigmentation in zebrafish and also in related species. So what we have in our model is U, which is melanophores, is repelled by V, by xanthophores, and therefore this interaction coefficient will be set negative. And on the other hand, V, the xanthophores, they are attracted by U, the melanophores, and therefore this interaction coefficient has to be positive. And the interaction range of all cells for all components is set to 0.1 because we don't know that much about it. And yeah, then you can simulate the model, and I said something about the heterotypic interactions which we assume here, but I haven't said anything about homotypic interaction, and at first we set them to 0, neither aggregating nor repelling homotypic interaction, and then the uniform steady state is just stable at t equals 0, uniform steady state plus some perturbation at t equals n. It has all flattened out. Also in this parameter regime, if you prescribe an initial pattern, the model is not able to sustain such a pattern, so it dies out to the uniform steady state. So for no homotypic interaction, we do not get any patterning whatsoever. So we have to put in some homotypic interactions, and here are 3 possibilities. In this plot, we have said that the U is positive, so attracting, and then what we see is we get mixed aggregates. So that's then probably not the right choice, because there would not be stripes, it would be grayish white and everywhere, and so this is not what we are looking for. Instead of mu UU positive, you could set mu VV positive, and then we get a pattern like this. Or we could set both positive, and we get a pattern like this. And from those two, this one is the more realistic one because it's persistent in time, and this one is yeah, here you have it's highly dynamic, and you don't see that, I think, too often with zebrafish, that they change their stripe patterns yeah, during the day. So with this assumption, we obtain a realistic pattern with our model. On the other hand, we have to admit that it is more likely that other factors are also important, in particular this iridophore cells, which should be taken into a model, and then maybe the problem can be understood even more. So this, that with only homotypic interaction and run and chase, that you do not get that any pattern. This has also been obtained by Thomas Woolley, who was here in the program in July, for a discrete particle-based model. So this is quite then in line with what we observe here. Okay, yeah, non-local models extend the non-local models I've shown here. They were originally developed for cell adhesion only, but they extend quite nicely from their original application to also include repelling behavior. And they capture important aspects of cell movement driven by direct contact. And we have applied them in a couple of contexts here. For cell adhesion, I haven't shown you simulations, but there have been in earlier talks this week. New request, dispersal pigmentation pattern we have seen. The outlook is, yeah, we have some quite good numerical scheme, efficient numerical scheme for the non-local term, but there are always possibilities to improve on that. A couple of analytic investigations would be in order, in particular for understanding traveling wave solutions, for instance, or boundedness of solution. And what we're also currently interested in is a relation to a suitable microscopic description, which then scales up to an equation like this. And this is work Thomas Hill and his PhD student and Kevin Painter and myself are working on at the moment. Thank you very much.
Any questions for Alf?
Please.
say a little about the time evolution from your initial conditions to these pattern states?
Well, it depends pretty much on the mu parameters. If you make them large in absolute value, then it's fast.
Because the question.
Or it's a different question you ask.
The question is essentially there's potentially linear instabilities which precipitate the aggregation. But then the question is whether there's slow dynamics which ensues thereafter, and you have the evolution of pattern dynamics.
That the pattern is changing afterwards, you mean? Well, you see this. Well, you here have this merging and emerging behavior. I mean, this is not explained by the linear analysis.
Sure. So it's the description of that, the extent to which you have a description of that slow dynamic process.
Oh, we haven't looked into that.
Okay, Jose and then Anatilla. Jose, on you.
How important is the diffusion, the diffusive time for the patterns? I would say probably not, no?
Probably not, no. Not much, although there was a recent paper by some Japanese colleagues which, well, exchanged our simple diffusion for a nonlinear diffusion term, and they got a much stronger separation of the peaks. But for us, we have put it in. Diffusion is kind of small, and we're solely focused on this interaction via contacts and the non-local term. But we could
Yes, if you don't put linear diffusion, you will have also in some cases complete segregation.
yes, I guess
You don't need nonlinear diffusion for that.
without if you have no diffusion at all, yeah.
This actually follows. So, can you comment on the comparison between the models and the Turing theory for pattern formation?
Well, this is more or less what you do for a Turing pattern.
But the first one is only a single equation. So
That's true, yeah. But you have with this.
What drives patterning? If I may ask, what drives patterning?
Well, the integral term.
Self-aggregation.
The integral term drives patterning. The diffusion.
There is no diffusion.
Well, there is just a little bit diffusion, but it's not important for the patterning. If you have, if you look at diffusion chemotaxis systems, you also get the patterning driven by the chemotaxis term, or the chemotaxis coefficient. And you either get a homogeneous steady state if taxis is maybe too small compared to diffusion, then you're sort of in the diffusion limit, or you get the pattern formation if your taxis is strong enough.
Yeah, the typically experimentalist question. Your theory contains some pretty abstract functions like this interaction kernel and stuff. How could, if one, I hope somebody is already working with you and trying to test this, what are the crucial experiments? How can one really test this approach?
Well, this is actually why we look at the microscopic model.
Yeah.
Sort of because in a microscopic model, what you put in there is much more closer to biology, and then when we can identify by going from the microscopic description to the mean field description, how this, for instance, this kernel should look like, and in fact, we also got some different kernels out of our current studies there. We can reproduce those, but you can also get different forms depending on what assumptions you make on the microscopic model, and that can then help sort of to better parameterize this model with biological data.
There is going to 2D. There is always, or there are several dangers that can happen. For example, these concentrations could not be extended as stripes, rather like bumps.
Yeah.
Have you looked into that or not yet?
But we've done 2D simulations, and you get sort of spot patterns or meanders, different.
For the zebrafish, you would like stripes and not spots, right?
Yes, but we didn't. I haven't done the zebrafish simulations in 2D, to be honest.
But run and chase doesn't seem to. This is the same question. Doesn't seem to. There's no obvious way in which that will give you stripes. It seems, unless being constrained by the fact that there is a boundary somewhere.
In 2D, yeah.
The Meinhardt models actually developed for the shellfish incorporate time in the evolution of the cell itself. So it is in effect one-dimensional. And as far as I know, it's one of the clear-cut cases, but only one of the few clear-cut cases where there. Yeah. Here might be.
Yeah, maybe you should look into 2D for that, but we haven't done.
Kondo's done some stuff where he looks at mutants where the melanophore-xanthophore interactions change, and then you get labyrinthine patterns or wider stripes and things like that. Have you thought about that at all? Whether you could
Well, as I said, we haven't done 2D for.
If you were just looking at wider stripes, you could do it in 1D. I mean, for labyrinthine patterns, you'd need 2D, sure, but.
Haven't done, but I mean, we have in these experiments, we've seen that if you change parameters, we get a different, like a longer wavelength mode is preferred. And
I think he had some intuitive explanation for why he felt the wider stripes are forming. You're somehow reducing the repulsive or attractive effects at the cell scale level, and it might be interesting to see whether you get that out. If you make the same assumptions he makes.
But the wider stripes might also be concerned with growing. Yeah, I think that's also.
Sure.
So you grow up to a certain width, and then you split into two stripes.
Sure.
So there might be different interactions.
If I understand, you only try, say, kernels that are they have a definite sign. Say, they are aggregating type of kernels.
Yeah.
Right? You didn't try something that is a repulsive part and an attractive part, like Leonard Jones and something like that.
No.
Okay.
Yeah. So to have sort of contact inhibition locally and for
makes
yeah, you had a plot like this in your
yeah, that's why I'm
in your talk. Yeah. But you haven't tried, no.
You also took the diffusion is dependent upon density.
Yes.
So what exact form you have selected for simulation?
Which exact value?
Exact form. D is a function of U.
what was the form of
What is the form? It is linear or non-linear?
Oh, you mean for this
That du.
that du?
Yeah.
Well, you can take some sort of like a Michaelis-Menten function, some saturating function going to or
You have taken something like.
well, I haven't done the. This was I haven't done the simulations right now.
But in
It's this is taking what other people have done in literature, at least this one here, to have sort of not a dispersal of your population. You have to have bounded dispersal. And not infinite speed of.
maybe linear.
Yeah, I think we need to do some lunch or taxes. So
Now, taxes might lead you to Churchill.
please thank Alf again for a really splendid talk.
Thank you.